Okay, good morning, all of you. So now we have another couple of minutes before we can take the today's speaker, Professor Alok Dhawan, who is also a host of this entire program. And exactly at 11 o'clock, we will start. And we have a few more minutes, a couple of minutes. And that will allow more participants to join from YouTube and Facebook. Just now we have started. So, a very good morning to all of you. Today we have Professor Alok Dhawan, who is one of the very important persons who is involved in conceptualizing this summer research training program and who is working very closely with our team to lead the program from the front. And upon our request, Professor Dhawan has kindly given his consent with a 24-hour notice to give this lecture. And we are very much looking forward for his lecture. And as a host, he and I are very similar, and I don't want to speak further, but for the benefit of a number of students, I request my colleague, Dr. Saurabh Barua, to introduce Professor Alok Dhawan. Now over to Dr. Saurabh Barua.
Good morning, everyone. Honorable Director, CSIR NIST, Professor G. Narahari Sastry. Today's guest of honor, the lecturer, Professor Alok Dhawan from IITR Lucknow. It is indeed a privileged moment for me to read out or to introduce Professor Alok Dhawan to all my fellow colleagues and students who are actively participating in summer research training program that has been hosted by CSIR, where your active participation has structured many eminent personalities to deliver, to encourage you all. And Professor Alok Dhawan, being a host, is one of them who has volunteered himself within 24 hours to deliver his lecture. Professor Alok Dhawan is currently Director, CSIR Institute of Toxicological Research, Lucknow, and Outstanding Professor, Biological Sciences, Academy of Scientific and Innovative Research, ACSIR. He served as a Director, Additional Charge, CSIR Central Drug Research Institute, Lucknow, which we call CDRI Lucknow, from December 2017 to July 2018. He was the founding director, Institute of Life Sciences and Dean, Planning and Development, Ahmedabad University, Gujarat. He has played an important role in building institutions of excellence in higher education and national laboratories across the country. He obtained his PhD degree in excellence in biochemistry from University of Lucknow, India in 1991, and was awarded Doctor of Science degree by the University of Bradford, United Kingdom in 2017. He has visited several countries both for conducting research as well as to deliver lectures at scientific meetings. Professor Dhawan started the area of nanomaterial toxicology in India and published a guidance document on the safe use of nanomaterials. His group elucidated the mechanism of toxicity of metal oxide nanoparticles in human and bacterial cells. His work has been widely cited. He has set up a state-of-art nanomaterial toxicological facility, which is a unique facility in CSIR IITR. He has also made significant contribution in the area of genetic toxicology. His work in the area of nanomaterial toxicology has earned him international accolades as well, and he was awarded 2 Indo-UK projects under the prestigious UK IERI program. He was also awarded 2 European Union projects under the FP7 and New Indigo program. As a mission director, he steered CSIR Mission Mode program on food and consumer safety solutions focused, involving several CSIR institutes to provide technological solutions from farm to fork. Apart from the national reference and referral centers of the FSSI AI, he has been instrumental in establishing BIRAG Bionist and DSIR CRTDH at CSIR IITR to promote entrepreneurship. Professor Dhawan has won several honors and awards, including the INSA Young Scientist Medal in 1994, CSIR Young Scientist Award in 1999, the Sakuntala Amir Chand Prize of ICMR in 2002, award lecture in the field of toxicology by the National Academy of Sciences India in 2008, the Vigyan Ratna by the Council of Science and Technology UP in 2011, Lucknow University Alumni Association Award 2016, Professor S.S. Katia Endowment Lecture 2019-2020, the Indian Science Congress Association. He founded the Indian Nanoscience Society in 2007. In recognition of his work, he has been elected Fellow, the National Academy of Sciences India, Fellow, the Academy of Toxicological Sciences USA, Fellow, the Academy of Environmental Biology, Fellow, Academy of Science for Animal Welfare, Fellow, Society of Toxicology, Founder Fellow, Indian Nanoscience Society, Fellow Gujarat Science Academy, Royal Society of Chemistry, National Academy of Medical Sciences, President, Society of Toxicology India, and Vice President, Environmental Mitigation Society of India. He has, to his credit, more than 150 publications, 20 review book chapters, 7 patents, 6 copyrights, and he has edited 9 books. Apart from this, he has kept one of the feather of CSIR in IITR Lucknow. And in this regard, we are really honored, sir. And I take this opportunity to invite you to deliver this lecture for the August gathering and for our SRTP students. Thank you, sir. Over to you, sir.
Thank you very much, Dr. Das, for this rather longish introduction. And I must congratulate Dr. Shastri and his entire team at NIST, who are taking this SRTP program from stride to stride, from strength to strength. And yesterday we had Dr. Inu Swaroop give a very exciting talk on STI policy and how she's been able to steer the program through DBT. Because she spoke about STI and how it has happened, so I thought I'll just change my talk a little bit And although I'll be talking about what it has been able to do for the country, but I thought I'll just share a few examples that will give you an idea as to what actually, when we say about STI or investment in science, technology, innovation, what it actually means. Does it only mean money, or does it also mean investing in human resource or creating human resource? So I think that's the pitch I'm going to make. So I'll just take a second and share my slides, and then we're good to go. Yeah, so Dr. Shastri, is it visible now?
Yes, yes.
Wonderful. So a very good morning to all the students and the mentors who are online. And once again, I wish to thank Dr. Shastri for giving me this opportunity to share my thoughts on what we envision when we talk about an Atmanirbhar Bharat, and essentially meaning that we are looking at a self-sustainable and self-reliant India. And when we talk about a self-reliant or a self-sustaining India, we also talk about, in the same stride, an India which is confident of itself, which has enough self-confidence and motivation to innovate. Because any country, when it moves forward, requires a lot of innovation, a lot of science and technology, so that the country move forward. And that's how the Western world has been able to fuel innovation. So, and when you talk about Atmanirbharta or you talk about self-reliance, I think the person who comes to your mind at the first instance is, of course, our beloved President, Dr. APJ Abdul Kalam, who showed the path of self-reliance to this country. In times when we were denied technology, he was the person who actually led from the front. And that's why India is where it is now. So I think before I move forward, just on a lighter note, I think during the Corona times, what has borne the brunt is actually the sports arena, and we are unable to see any of the matches going on, be it cricket or football or tennis or anything. So I thought I'll just so that students remember some of these words. BCCI everybody knows because of cricket. But when you talk of an Atmanirbhar Bharat, when you talk of a Bharat which has to become self-reliant or confident, I think what I would say for BCCI is to be critical, creative, and innovative. So I think this is what the students need to remember at all times. The second that I mentioned is the Kabaddi League, the Indian Kabaddi League. But here what I mean is the Indian Knowledge League. This is what the students need to create. Knowledge league for the country, and that is what is going to take us forward into the next decade. And that's how we are going to become self-reliant. And of course, the IPL, the most coveted IPL that everybody watches on the television so fondly, is the intellectual property leader. So that's what the audience as of now on the digital platform needs to become, intellectual property leaders. So I think that's what my message would be to begin with, that we need to create an Indian Knowledge League and we have to become intellectual property leaders. Only then our country will become Atmanirbhar, and only then we'll have the self-confidence or Atmavishwas. I think before I venture into how we can become Atmanirbhar or how we can have self-confidence, it is also important for us to understand how we think differently or how we sort of conceive ideas differently. So, for example, Council of Scientific and Industrial Research is the name of CSIR, where industry is right in the middle. But at the same breath, I would also like to mention that it is also a citadel of scientists for innovative research. So I think this is what I think about CSIR. And I think it's very important that each of us think of CSIR in a fashion that we are able to contribute to this, not only to the organization, but to the country as a whole. At the same time, I was thinking about CSIR and what we sort of inculcate in our students or among the faculty or the culture of CSIR is to collaborate, cooperate, and co-create. I think that is the mantra that CSIR always fosters. Not only that, we also believe in a SKY, which is with a difference. So SKY is the normal spelling, as we mentioned. But I keep saying that SKY is what you can see. So if your goal is the sky, and that's what we say, but in vernacular, you say pratyaksham kim pramana, meaning that if you can see something, you don't need evidence. So if you can see the sky, you perhaps don't need evidence. But if you spell that sky with a difference, that is science, knowledge, and innovation, SK and I think that will be the road to excellence as far as we are concerned, and that's again something that we foster in CSIR. And third, of course, is I stands for innovation. All the time we innovate, and innovate. And the fourth is risk-taking. So that's another advantage that CSIR as an organization, but the country as a whole has been able to do over these several decades post-independence, is to provide enough risk mitigation, provide enough opportunities for scientists and youngsters to take the risk so that they can take the science and the country forward. Just so that you understand the SNT ecosystem, what we were able to show in this slide is that CSIR was among the first organizations to be started even before independence in 1942. And as we move forward, different organizations came to the fore, adding another dimension of science and technology and innovation into the entire ecosystem. And last but not the least, DBT came in 1986, and you heard a fantastic exposition of what DBT is doing by Dr. Renu Swarup just yesterday. As far as CSIR is concerned, we are a society which is headed by the Honorable Prime Minister of India, who is the president of CSIR. The Vice President of CSIR is the Honorable Minister of Science and Technology and Earth Sciences. And we have 38 labs spread all over the country. And in Lucknow, we have 4 laboratories. CDRI, which is the Drug Research Institute, CMAP, the Institute of Medicinal and Aromatic Plants, our institute, the Toxicology Institute, and NBRI, the Botanical Research Institute. So that is the length and breadth of CSIR institutions across this country, and that is how we are fostering science, technology, and innovation across the board. If you see this graph, which clearly depicts that the era of 30s to 50s was that of individual excellence. Then came the self-reliance, then technology denial-driven technologies were developed in our own country, and then we came into an era of IP generation and global competition. And finally, we are looking for global SNT leadership, which is quite apparent and evident from the kind of technologies and publications that are coming out of CSIR and other research organizations from this country. And this country has tremendously improved in the quality and quantity of science that is being done, as well as the technology. So we are moving towards a path of Atmanirbharta and self-reliance. At the same time, we are also not losing sight of the global SNT leadership. CSIR, right from the very beginning, if you see, was aligned to the mandates of the country, and right from the very beginning, if you look at the indelible ink that was required for conducting elections, all the way to milk powder or the tractor or pesticides or medicines, planes that we have developed, so on and so forth. So I think over the period of time, we have shown that CSIR has the capability to nurture science, technology, and innovation and take it forward to self-reliance in this country. I think CSIR by and large is divided into these different sciences: physical, chemical, biological, engineering, and information sciences. And due to which we are able to not only galvanize different sciences into solving a problem, but the other advantage being that, as I mentioned, we can cooperate, collaborate, and co-create within CSIR, and of course outside of CSIR, because we have the bandwidth in all the sciences that are there in this country. More recently, if you look at the mitigation strategies for COVID-19, CSIR took the lead very early on because that was the need of the hour at that point in time, and the entire CSIR got galvanized into looking at the this challenge and seeing how we will be able to solve these issues that are facing the country. And for that, CSIR made 5 sort of verticals, one for rapid and economical testing. The second was for digital and molecular tracing. The third on new drugs, repurposing of drugs. The fourth on hospital assistive devices as well as PPEs. And the fifth was supply chain and logistics support systems. And if you look at this, the logos, you will find that already CSIR was, as is true to its name, was working closely with the top industries in the country. And we were very fortunate that they have actually bestowed their trust on the quality of technologies coming out of CSIR. And it was very nice that within a very short span of time, CSIR was able to deliver on various counts, and all the products that have been developed are either in market or in the process, and the technologies have been transferred. So that was a showcase of the strength of CSIR when you talk about science, technology, and innovation. So I think it should give the students a great amount of pride that they are associated with CSIR as of now in various institutions where they can actually look at the kind of work being done, and in particular, of course, interact with their mentors. But I think this showcasing by various, I would say, thought leaders in science that is happening through these lectures, I think is phenomenal for the students to get an overview of what is happening in these various organizations or institutions. Last year, an India Innovation Index was released, and since I'm from Uttar Pradesh, I thought it would be prudent to also mention that we stand at number 7 in terms of innovation, and that's something we need to see how we move up the ladder. Karnataka is number 1, Tamil Nadu number 2, Maharashtra at number 3. So I think, again, this is something that should give us enough energy and enthusiasm to see how we should move up the ranks and ensure that we sort of foster innovation, and not only innovation, but also entrepreneurship. Coming to my institution, which is the Indian Institute of Toxicology Research, I must mention that this is the only institute of toxicology in this country, and there are a handful of the toxicology institutes in the world. So it is a very unique institution. That is one. Second, I would say that the very definition, by very definition, Toxicology Institute is a translational institute. And the third would be that the toxicology actually defines the domains of safety. Safety per se does not have any domain, and toxicology provides a framework by which we can sort of define the domains of safety. Right from the very beginning, the motto has been safety to environment and health and service to industry. So what the need of the hour is that we should all work for the common man and for the industry. This institution has been doing for almost 55 years, since inception in 1965. And we have been fostering innovation and good quality science and technology in this regard. Currently, our portfolio is in 4 verticals. The first, of course, basic applied and translational research in toxicology. And very importantly, the research for policies that we are doing. And that permits us to feed into the government policy making bodies what are the requirements for policy. The second is equally important, is to assess the safety of various chemicals and that are there, or drugs and pharmaceuticals. And for that, we are enabled or certified both by NABL and GLP, which is a good laboratory practice. The third is human resource development. Again, a very, very important aspect of investing in STI. We need to create good quality human resource in various areas. And I must mention that I think today is the 5th year of the skill development program run by the Government of India. And we are very proud that we also run a lot of skill development programs across the length and breadth of CSIR. And so does IITR also run a skill development program. And last but not the least, we also foster linkages between entrepreneurs and industries, and of course academia, through the Center for Innovation and Translational Research. And this allows them to actually come and work with us and create and co-create things that are important for the government, and we'll share some thoughts on that as well. These are the Navratnas, as they call, of IITR, and we work in various areas, including food, drug and chemical toxicology, systems toxicology, environmental toxicology, nanomaterial toxicology, computational toxicology, research for policy, and of course we do service to industry, and all this that we do has translational research at the core. So that's how this institution is sort of built. Now I'll give you some examples of how the innovative thinking of our scientists has transformed certain areas. One of which was, of course, safe and clean drinking water. That again, if you look at the CSIR portfolio, you'll find right from desalination to portable technologies, everything is available within the CSIR fraternity. Several of those are now commercialized and are in public space. What we thought is look at a device that could disinfect, because by and large water, when it is pumped in the municipal supplies, is good, but we need to disinfect this water when it comes to the tap. And so for that, we developed a device which was membrane-free and chemical-free. And what we were able to do is to disinfect water within no time, if I would use that word. And both a household model and a community model were built, and both can run on solar power. So that was the thought, that when we interacted with the stakeholders, what they want, the main concern was that once we put up a sort of plant at home or a water purifier at home, there are a lot of consumables that are required, and every year we have to pay a hefty amount of money for the ANC. So now the companies who have taken the technology, at least for the community one, they are promising 10-year sort of warranty, as well as they are saying that you don't need to worry about the maintenance, because it is a IoT-enabled device and it can inform you as to whether something is going wrong or not. So with this, we were able to develop, as I mentioned, the community system device, which is 450 liters of water per hour. And I'm very happy that this has now gone out into the market, and our technology partners are actually supplying it. This was something which was very innovative in that sense, that we could provide clean, I would say, disinfected water, primarily because the waterborne infections are very high in the villages as well as elsewhere. So we thought it would be a good idea to look at this. So I think this was very well received by the policymakers as well as by the public. As I mentioned, we have now been able to transfer it to two entrepreneurs, one of them being a startup. And the startup is from Lucknow itself. And we are able to sort of not only empower the startups, as I mentioned, if you talk about science, technology, innovation, and Atmanirbhar Bharat, we need to ensure that the startups are empowered. We need to ensure that the scientists who are doing the work also get motivated, because they can see the technology going out to the market. Another technology transfer was done by the Honorable Minister himself in Delhi, and both these companies have now been able to make the commercial prototypes, and they are now available for public to purchase. As far as the as an institution, you must be aware that we were also looking for what we can contribute when the COVID-19 situation arose. And so therefore, in alignment with CSIR's 5 verticals, we started looking at what all we can contribute to the national cause. And I'm happy to inform the House that we are not only looking at the sequencing of the virus as part of the CSIR consortium, but we've also created a rapid and economical test facility at IITR for testing COVID through RT-PCR. And just this week, we completed 20,000 tests for the patients, the samples of whom we get from the state government. In the other vertical, we are also looking at repurposing of drugs. Again, the scientists in computational toxicology are working in this area to look at how we are able to repurpose the drug, but also reduce the toxicity, looking at structures and trying to help other institutions among CSIR to develop the drugs. When this Corona or COVID started, the first call that came to us from the government was for sanitizers, whether we can help the government provide sanitizers, and this is what our scientists again stood up to the challenge. And within a day or so, we were able to provide a substantial amount of sanitizer, hand sanitizer to the government so that they could open their shops and start their work normally. This was when the first lockdowns happened. And subsequently, we've been offering this to the government on various occasions to the various people who are in the forefront. And I would say when you invest in science, technology, innovation, you are investing in knowledge for the future. And I think these institutions of excellence and eminence that have been created have stood the test of time and has also stood up to the national crisis of COVID-19, wherever they were in the country. And I'm very glad that again, a startup came to us and they actually wanted to start manufacturing this hand sanitizer, which they did. And again, this whatever we were able to sort of have was transferred to a company, and now they are actually manufacturing and selling it themselves. The third, this again is a good example of how we work jointly and co-create systems. So there was a startup working with us earlier on, and they had a microwave device which can disinfect biomedical waste. But they came to our institute to work with us to see what other applications can be built or inbuilt into this device. And that point in time, we had looked at how we can disinfect linen in the hospitals, especially in ICUs, where there are people who are infected by various diseases. So we looked at the linen disinfection. And when COVID-19 came in, again, that startup approached us and said, we'd like to see how we can convert that linen cycle or other cycles into disinfection of PPEs. And ultimately, they were able to come up with a cycle that can disinfect using microwave, the PPEs and N95 masks. So now, currently, we have been able to undertake the experiments, and the results have been shared with the government so that they can take a call, so that they can use it. And this was again something that was very well received by our government. Both the Honorable Minister tweeted it, as well as it was up on the Mann Ki Baat updates, a Twitter handle used for by the Honorable Prime Minister, where the new things that come out and which are contemporary are also highlighted. So again, it shows that if you're sort of creative enough and innovative enough, we can do wonders. And this was all an effort towards ensuring that we do not have to import things. Even if you look at the APIs, there were a lot of things that we had to import all the time, but our CSIR Institute sort of stood up and they were able to develop processes that can now be, that are able to indigenize the production of APIs, the active pharmaceutical ingredients. That has helped our pharma sector in a big way. And the fifth vertical was the supply chain and logistics support system. So when you are developing devices, etc., when you're doing tests, you need to understand as to what will be the supply chain, where are the things available. And initially, as you might be recalling, there were a lot of issues in procurement. So that's where CSIR again chipped in, and they developed a portal for supply chain management and logistical support. And IITR is one of the nodal sort of institutions for the North. And we are again participating in this program of supply chain management. So all the 5 verticals our institute is participating. So when I say that we should invest in STI, I think these are times of crisis for the country, and whenever such times arrive, I think the scientific institutions should stand up and ensure that whatever it takes to sort of mitigate this problem, this should be done at the topmost priority, and which CSIR has shown that it was able to do in a very short span of time. So I think my heartiest compliments to all the scientists, the students, the technical staff of CSIR, and of course the leadership of CSIR that stood to the test of time. Now I'll share a small story with the students so that it gives you an idea as to how when you're faced with a challenge, you need to sort of stand up to it, you need to ensure that you have a positive thought process, and how I should say, in a sense, that we are fortunate that over the decades, people have ensured that the science, technology, and innovation paradigm has been supported to the hilt, if I may use that word, in this country. So this is a small story that I'll share with you. This was 2005. I was in Michigan State University, and the 2 gentlemen that you see here are from the chemical engineering group, Professor Hasham and Professor Tarabara. And it was a very cold winter day when we had a meeting that was perhaps on a Friday. And they asked me that, those days they were synthesizing nanoparticles, especially fullerenes in their department, and they wanted to see what is the toxicity of these fullerenes. And I must mention here that at that point in time, although our country had begun synthesis of nanoparticles, colloidal particles were being done even earlier, but the toxicity or safety assessment was not there in the country. So the first thing, as a toxicologist, I spoke to them and I said, well, can I come back to you on Monday and we'll discuss? Because I also wanted to understand how to undertake this toxicity. And I think at every point in time, we should be open to learning. I think that's a very important facet of the STI policy, that if you don't reinvent yourself, keep learning, and you don't sort of change with time, it will be very difficult even if we put in a lot of money. So I think money, of course, is one part that we keep investing, but I think at the same time, it is incumbent on the scientists to ensure that we keep doing things which are new, novel, but at the same time, keep track of timelines. And what happened next was that I actually wrote to the then director of my institute, asking him for permission that I could do some toxicity studies. And this is again something that I never stop admiring, that I wrote a long, lengthy email telling him about the importance of the nanotechnology that is coming up and how toxicology will play a major role in the technology coming to the fore. And if you look at his response, it just says, please go ahead. I think that is the spirit with which this country is going to become Atmanirbhar, and that is the spirit that we see in today's days and times when people are supported even at the grassroots level by various organizations, by various government schemes, that we are able to uplift the people even from the grassroots, even from the villages. And they have, at least from CSIR point of view, I can share with you that CSIR ran a program through which we were able to capture sort of innovators from rural backgrounds, and they went all the way to the CSIR Foundation Day last year, where the Honorable President actually gave away the awards to them. So I think this is how we will be able to foster innovation in science technology and make this Bharat full of Atmavishwas, self-confidence, and self-reliant. So as soon as I got this reply from the director, I put my head down. We did some studies, and I'm very happy to state that, please see the timelines. We are 2006, we had a publication out. Perhaps the most highly cited article for these 2 gentlemen whom I showed. But at the same time, it was another area that I was getting into, which was nanotoxicology. And not only that, it's a very, very highly interdisciplinary area. So as soon as you start investing in STI, you also have to ensure that at each step, we are able to rope in people who will be important for taking that innovation or that sort of technology forward. It doesn't matter which field of science they come in. So I think this was a brilliant example in almost 15 years ago of a collaboration between environmental engineers or chemical engineers and toxicologists to sort of resolve an issue that was pertaining to the colloidal dispersions of C60 fullerenes. As far as nanotechnology is concerned, you are all aware that it is an enabling technology. And the issue for toxicology was that on the one side, you see nanoparticles which are almost 1 nanometer in diameter. On the other side, you see DNA which is 2 to 12 nanometer in diameter. So I think apart from the chemical interaction, they can actually physically interact with DNA and other macromolecules within the cell. So I think that was the sort of concern globally that was there. So with that in mind, we started looking at how we should assess the nanotoxicology in this country. And again, to simplify the issue, if you look at a sphere, or if I may use the vernacular again, if you look at a bundi laddu and you take out the bundis separately, so instead of one surface area, you get a huge expansion of the surface area. So when the surface area increases, the reactivity also increases. And as you go down the size, the sort of molecules at the surface increase. So that's how nanotechnology, when you break down the particles from a normal size to nanoparticle size, at times they create toxicity or safety issues. So these were things that we highlighted at that point in time, and we moved forward the various issues that were pertaining to the toxicity of these materials, and each of them were sort of addressed at the institution along with collaborations from IITs and other CSIR institutions. And we looked at the various approaches, could it be in vitro or in vivo or in silico. All of these approaches were looked at to ensure that whatever we are doing can be done in a time frame, and the outcomes should be sort of good enough so that the world can take cognizance of the fact that whatever we are doing or the data that is being produced can be utilized. So the first thing, of course, when you talk of science, technology, innovation, is to look at what are the tools at hand. So, of course, you may be aware of the electron microscope, which can be used for looking at the nanoparticles, but it's very, very cumbersome to prepare a sample and look at the nanoparticles and, of course, the cells, etc. So we thought, is it possible that we can use a flow cytometer, which is a much simpler device, and the amount of cells that can be seen are in several, hundreds of thousands. And is it possible that we can look at whether the nanoparticles that are getting inside the cells can be monitored? So what we actually did, based on the principles that when you get nanoparticles inside the cell, the side scatter will get increased. If they are stuck outside the cell, the forward and side scatter both will increase. So these were the basic principles of the flow cytometer. And so using those principles, we first look at the uptake using electron microscopy, and you can see these black dots, which are essentially the titanium dioxide nanoparticles inside the Hg2 cells using electron microscopy. But at the same time, we looked at these cells and whether we can monitor these uptake of nanoparticles using flow cytometer. And we found that it was quite good in terms of monitoring the uptake of nanoparticles in human cells. Similarly, in bacteria, and here I'd like to draw the attention, especially of the students. This was a fantastic observation that one of my students actually made, that while these bacteria was dividing, you still have nanoparticles inside them. So the student said that is it possible that we can actually look at various generations of bacteria and see whether the nanoparticles are still there or not. And again, we looked at first the internalization. Now you can imagine the size of bacteria and then the size of nanoparticles going inside it. So first, of course, we looked at the electron microscope and then flow cytometry. And we were able to show that this method of flow cytometry is very good to see whether the nanoparticles, especially metallic nanoparticles, can actually be monitored or the uptake can be monitored in bacteria. And then we looked at the multi-generation study, and up to third generation, we could actually undertake these studies and find nanoparticles are still inside after the first sort of exposure. And of course, then we took on to our in silico approach, which again, I feel these are some examples that I'm showing you. Again, we looked at the graphene oxide and a alpha helical protein. So you see the hydrated systems where the protein is able to survive. On the other side, we took graphene and the protein over a period of time actually lost its alpha helical sort of structure. So this was a biology problem. We used computer sciences to solve it, and it was published in a chemistry journal. So I think again goes to show that your innovative thought process and the fact that you're able to understand the mechanisms, what are the tools, is very important to solve problems. Because of which we were able to actually come up with various facets of engineered nanomaterials, right from synthesis all the way up to prediction and toxicity at this institution. And we now have a national nanotoxicology sort of facility, which has been provided to us by CSIR. Now, when you talk of this kind of work, I always feel that it is important to see what impact it makes globally. So this was a publication we made on the DNA damaging potential of zinc oxide nanoparticles in human epidermal cells. The timelines were that it was available online in January of 2009. In April of 2009, the Science for Environment Policy of the European Commission had picked up this paper, and they actually wrote about this paper a full page, where they mentioned that these kind of studies are needed to ensure whether we can use nanoparticles in cosmetics or not. And currently we are having more than 580 citations for this paper, and is one of the most cited articles of the journal. So I think in the country itself, we have enough scientific grit and determination and the capability of students that they can actually perform studies that are globally relevant. Not only that, the flow cytometry method, which I described to you in the nanoparticles uptake in bacteria, became one of the highlighted articles in the Cytometry A journal, which is to do with novel methods. So again, if you think differently and you're innovative in your approach, this is the name of the student, Kumar. Now, essentially, he has a method in his name forever. So I think, again, these are things that students should remember. Don't take the easy route out. Try to spend some time, solve a problem with innovation. Right? And the third was, of course, the Langmuir paper that I mentioned to you. And I just share some more slides, and then I'll perhaps close for question and answer. But here are the timelines. As I mentioned, the first thing that I had done in November was to shoot out an email to my director in 2005. When I came back in February, we started establishing the facility in 2006. We had then submitted by December 2006 a project with NCL Pune, which is again a CSIR lab. Established the Nanoscience Society in 2007. We did an international meeting in 2008. Then in the meantime, we had got the UKRI projects, two in succession. We had a European Union project with 8 countries, and this was remarkable because we were sort of teaching those countries how to perform nanotoxicology, and they actually came here and got trained and went back. CSIR also started a program on this safety of nanomaterials. We were also awarded FP7 project, NanoValid. This was one of the flagship projects of European Commission, in which 18 to 19 countries actually participated. We did another symposium in 2011 where 15 countries participated. All this was being done so that we can generate enough human resource and try to understand what is the best in the world in this area. We published a guidance document on the safe use of nanomaterials in 2011. 2016, an advanced imaging facility was inaugurated by the Honorable Minister for Science and Technology, Dr. Harshvardhan. And in 2017, when we looked at the Web of Science, CSIR was globally positioned among the top 5 in nanomaterial toxicology. And later on, I would say that in succession, our scientists have published 3 edited books in 2017, 18, and 19 on the various facets of nanotoxicology and their interaction through the Royal Society of Chemistry as well as the CRC press. This is the guidance document that we publish. So whatever you do, we should ensure that if it has a larger interest in terms of the policymakers, we should share it with policymakers. And once this came out, then the DST came out with a policy in which the institute participated on the guidelines of best practice of safe handling. More recently, in 2019, the European Commission actually has cited some of our references in the guidance for safety assessment of nanomaterials in cosmetics, as I mentioned to you earlier. But this is a document that has come out in 2019. The Honorable Minister for Science Technology actually released guidelines for evaluation of nanopharmaceuticals in the country. Again, our institute has been closely associated with the formulation of these guidelines. And it is indeed a very proud moment for all of us that now the country has a guidelines for evaluation of nanopharmaceuticals. Again, it will fuel the innovation cycle among the pharma sector so that they can bring out new and improved pharmaceuticals in the country. Not only that, just on the July 7th, again, this was a guidelines that was published and released by the Honorable Minister on the evaluation of nano-based agri-input and food products. Again, our institute has been instrumental in providing the inputs to these guidelines. And this is just a snapshot to show you how we have trained the faculty and students from 8 different European countries, and they went back and established their in their own countries nanotoxicology. So in a sense, we were able to train manpower for the European countries, and they've actually benefited from the investment that was made in this area by the government of India. As I mentioned to you, CSIR is positioned among top 5 in the world in 3 areas of toxicology: industrial toxicology, food, and nano. So I think the investments that have been made are I would say, they have provided fruit now, and it is very heartening to see that we are benchmarking ourselves to the global standards now. When you come to sort of looking at the investment in science, technology, and innovation, I think that is the only way we can take the country to self-reliance. That's the only way we can take the country to the top of all the countries that are there in the world. But at the same time, we should ensure that there are policies that will feed into the STI policy to ensure that all the stakeholders are able to benefit. And we should not lose sight of the goal that we need to achieve. That's very important, especially for the students, that when you have a goal in mind, I think nothing else should bother you. Just hard work needs to be done to achieve that goal. Again, for the students, I must mention that when you are into science, you need to have an eye for observation. So the same sample can tell you different things depending on the in-depth observation that you make for the sample. And here is a case in point where I'm looking at a photo of the child. But at the same time, if you are doing an in-depth sort of examination, you can actually see the person who clicked the photo inside the eye of the child. So I think it depends on the in-depth observation that you make. Another example is here where you have 2 birds. One is sitting here, the other one here is pretty weird, to be honest. So you magnify it and you still find it is very difficult to identify the bird here. But when you make a close observation, you'll find it's actually the crow with the beak here and the feather here and a ray of light coming through from the feather. So it depends on how you perceive things, how you observe them, what you make out of it, what are the inferences you draw, and how you take the next steps in your plan of action or where you have to go. So I think it's very important for all of us to keep that in sight. I have been indeed very fortunate. The work that I did for nanotoxicology also invited me to begin the TEDx Lucknow chapter, where I was invited to give a talk on how the sort of investment or the risk that was taken by the then head of the institution has resulted in the fact that CSIR now enjoys the topmost place in the world, among the top 5 people in the world in terms of nanotoxicology. So I once again, I must say that I have been very fortunate that CSIR has supported the initiatives or the sort of programs that have been initiated by me. And not only that, I would say the government has always been very clear in its mind that even if you look at Jai Jawan, Jai Kisan, Jai Vigyan, Jai Anusandhan, at every point in time, science, technology and innovation has been at the fore. Even when you talk of Jai Jawan, of course, we salute our armed forces, but to keep them up at the borders, there's a lot of science behind it. Again, we are also saluting the science that permits these Jawans to be at the border and be safe. You talk of the Kisan, again, a lot of science goes into it, a lot of innovation goes into the farming sector. CSIR has contributed tremendously in this area, and again, we salute the farmers. At the same time, we ensure that the science behind these people are the best quality that is available in the world. And of course, successive prime ministers added Jai Vigyan and Jai Anusandhan to it. And we are really proud that the science, technology, innovation is taking the forefront in this country, and all the scientific community is being looked upon with great reverence and of course with great hope that we can provide the solutions to the problems. This tree is the tree of our institution, where we look at not only different programs, but we are also fortunate to have the first bionest of BIRAC in CSIR, but also CRTDH from the Department of Scientific Industrial Research, and the Food Referral Center and the Reference Center of BCCI. So this again fosters the innovation, fosters technology development under the Technology Development and Innovation Center, which is being developed at the institution under the CSIR umbrella. And I must once again thank my dear friend Dr. Shastri for giving me this opportunity to share some things about what STI has been able to do in terms of CSIR, in terms of this institution, and how it was able to foster linkages not only within the country but abroad, and ensure that we get the best of the STI in this country. I think this is the only way forward for our country if it has to become Atmanirbhar or self-reliant. And I urge all the students to ensure that you should be encouraged and you should take the leap of faith to come into science, technology, come into research. And that's how our country will become self-reliant, and you'll take pride in saying that you don't have to go to a foreign country to do the science that you wish to do, and you can do it right here in this country, in one of the national labs, or the universities, or the IITs, anywhere. So thank you very much indeed, and I'll be happy to take any questions that are there. So.
Thank you very much,
Thank you very much
sir,
for everyone. And I request our people to take forward the question answers. Lucky Sakya and the others will take over. It's a wonderful presentation, and thank you. Lucky, you please take over.
Yeah, yes, sure, sir. Thank you, sir. Actually, there are a lot of questions, and we are getting a lot of appreciative messages regarding your talk. And we have with us our students, Miss Lisha, Kalita, and Monty. So now they will ask the selected questions because there are a lot of questions, and as such, I hope you will like to answer all those things. So now over to Lisa.
Thank you, sir. So, sir, the first question is from Mihir. He is asking how can we use nanotechnology for the drug delivery and what will be the challenges of these in future?
So, Mir, very good question, I must say. I think a lot of work is being done within the Institute of CSIR. Various institutions are working on this. They're using different nanoparticles as a carrier for a drug. At the same time, they're also trying to look at the possibility of using the drug and essentially making it nano size so that it can be delivered to a particular organ. Two aspects of that. One, of course, is to reduce the toxicity and take it all the way up to the organ that desires it. So you can reduce the amount of drug that is to be given. And the second is to reduce the toxicity aspect, especially in the cases of cancer. So I think nanotoxicology or nanotechnology is being used extensively, not only within CSIR, but also in other institutions, both in India and globally, to undertake. The challenge here is to see, because the nanoparticles actually cross various barriers, such as the blood-placental barrier or the blood-brain barrier. So the challenge is to see that the nanoparticle that you're making for drug delivery or the agent that you're going to use for drug delivery is safe enough and either does not cross the blood-brain or blood-placental barrier. And if it does, I think it should be absolutely safe to the brain or to the baby. So I think these are the challenges that we are facing as of now. But there are drugs that are already available in the nano-sized format.
Thank you, sir. The next question is from Tanu Atriwal. She is asking, is there any way we can use nanotoxicology in leaching out bioplastics? Can this method be utilized in degradation of other biodegradable materials?
So let me first clarify, Tanu, that nanotoxicology is a discipline which assesses the safety. Okay, once you assess the safety, then you look at how you can develop a method to either degrade the plastic or to look at microplastics. It is a way, it is a tool that you get to sort of look at how you can degrade the microplastics. We are doing a lot of work on pesticide degradation. We have been able to identify microbes that can degrade pesticides and that can degrade polycyclic aromatic hydrocarbons. We are still working on some of these aspects in terms of microplastics. We have not been able to get a particular microbe that can actually degrade microplastics. But nanotoxicology per se, if you ask me, that is a field that allows you to assess safety on nanoparticles. And based on that, you can decide the next course of action. I think that's how it is. Yeah, over to you.
Thank you, sir. The next question is from Suman Joshi. He's asking, enormous research globally on nanoparticles and their applications are going on. And you were talking about toxicology of nanoparticles. So how are you dealing with toxicity of nanoparticles?
Yes, I think again a good question, and that's why I sort of thought it would be a good idea to share with you how these particles become toxic and what are the principles. And when I say toxic, it doesn't mean everything is harmful. We also provide you a range within which the particles are considered safe or they are safe to use, and beyond that range it becomes toxic. So I think every material or every chemical that is being used, there is a certain dose, a certain concentration at which it can be used safely, beyond which it becomes toxic. So that is what nanotoxicology allows you to do. It allows you to figure out whether the concentration of the particles while retaining its efficacy, while retaining the application that it is meant for, can be safely used or not. I think that's the whole point that I was trying to drive. There's no way that I'm saying that don't do nanotechnology because it's going to be toxic. No, everything in this world that you deal with at some concentration or the other is harmful. Okay, what I'm trying to say is that based on these principles, we are able to develop a safe to use material and essentially ensure that you still have the efficacy for the application that you want to.
Thank you, sir. The next question is from Pooja Patel. She is asking, how can we make membranes using nanotechnology for all types of wastewater?
Well, then, what I think is, Dr. Shastri still online? Dr. Shastri not online? Okay. So what people are actually doing are trying to see if they can impregnate the membranes with different types of nanoparticles that are specific for a particular contaminant that can be removed. That will improve the quality of membrane if it is impregnated, and also improve the quality of removal. So membrane can last longer, and it can also be utilized. So that's how people are utilizing the nanoparticles. The other way they are utilizing nanoparticles for removal of contaminants is by taking nanoparticles, putting them in a cartridge and trying to push through the contaminated water or whatever they want to clear. And that's one of the technologies that has actually come into drinking water as well, where people are using cartridges, using materials which are nano-sized. They are part of that cartridge, which allows them to sort of take care of the pesticides and other contaminants that are there in water. Yeah, over to you.
Hello, sir.
Yeah.
Sir, the next question is from Tanuja. Are the guidelines for nanotoxicology is being followed in the area of cosmetics? How one can ensure of it?
So I think the guide, whether they are being followed or not, is the question for the company who is manufacturing, right? So I think there are regulatory bodies who actually look into these aspects while they are granting licenses, and that is why we came up with the nanopharmaceutical guideline as well. So that now if the people have to develop any nanopharmaceutical, they can do so with these guidelines in place, so that the regulation of the nanoparticle use can also be done. Same is true for the nanoparticles in the cosmetics. The regulator will actually ensure that they are being complied for. I think that should answer her question.
Thank you, sir. The next question is from Shivali Sharma. She is asking, can we use nanoparticles to make spray for food safety?
So it depends on as of now, we are not encouraging people to make sprays for food safety using nanoparticles. It will depend on the particle that you want to make. We have to look at its safety. We have to look at the risks involved, both in manufacturing as well as in spraying them. It depends on the particle. If it is a totally biodegradable particle that is going to provide safety to the food, we'll have to look at the safety aspects, and only then it will be permitted by FSSAI to be taken forward.
Thank you, sir. The next question is from Vaidehi Shripat Pisu. What is the role of toxicology in everyday life? Kindly guide for the research areas in toxicology.
Oh, that's fantastic. I think if you get up in the morning, the first thing you do, perhaps is you brush your teeth. So even the toothpaste has to be looked for its safety and toxicity. Anything that you use in common day, during the daytime, has gone through the portals of toxicology. It is the science that, as I mentioned to you, ensures safety of products that come to the market. And so therefore, it has a huge role to play as far as safety of humans is concerned. You also have occupational toxicology, where you look at the various occupational settings in various companies. You look at how it is, if at all it is harming the human beings, and we tend to then provide corrective measures to them, and then of course guidelines come into play, and that's how it grows. As far as the futuristic sort of areas are concerned, one thing of course we are looking at is how we can reduce the use of animals in toxicology. So we are looking at how to print 3D sort of organs on which we can do testing, or for example, can we have chips that on which we can do testing or understand the mechanism. So I think we're trying to see, and in particular, if you talk of areas, I think the nanotoxicology has a great future. We are also looking at the omics as the future of toxicology, and as we are moving ahead in the newer areas in chemical sciences and in material sciences, we are also looking at how to address or how to find methods to be able to address these challenges, where we are talking of very minute quantities of the materials in, let's say, water, or in humans, where we need to test. So is it possible that we can have sensors or devices that can test for you, and you don't at the point of care, and you don't need to sort of go out there? So I think we are also trying as an institution to work in those domains which are very highly interdisciplinary, and try to develop some of these devices that can check the contaminants for you as a handheld device or a wearable device, or whichever. So that's the future. I think that's where we are headed to.
Okay, sir. So the next question is from Mohit Mehta. He's asking, can education play any role to maximize the number of intellectual property? If yes, what kind of change do you think as a student I should do in study?
Fantastic. I think I must compliment, what was his name again? Mehta, some Mehta. Mohit Mehta. So I must compliment Mohit for bringing this point up. I, for one, have been always saying that the education system needs to be changed. We need to ensure that the students start thinking rather than just learning what they are taught. Once they start thinking, they will start understanding. Once they start understanding, they can innovate. So I think even now, since Mohit is perhaps done his undergraduation, that's how he is there, part of this program. We should try to see any phenomenon and try to think why it is happening, try to understand why it is happening. Only then you'll be able to take up new challenges, only then you'll be able to innovate. So for innovation, you should understand the basic principles. You should understand each and every step in that process. And I think that will be very enlightening, and that's what we feel, that in our education system, experiential learning is very important rather than just by books. So I think this is what the government of the day is also trying to foster, to ensure that the students get an opportunity to learn right from school levels. You have Atal tinkering labs now in school. You have other such programs that are there in colleges and universities. And so I think, Mohit, the fact that you started thinking in that direction already shows your interest in innovation, and don't let it die out. Be creative and start, find out your place where you want to work and just go ahead.
Thank you, sir. So the next question is from Dr. Meera Parmekar. She's asking, are the facilities at IITR open for other researchers across country? If so, how can we approach the nanotoxicity department if we want our samples to be tested?
Yeah, so the first thing when you say whether they're open, 100%, it's a big yes. Everything is available online. You can actually book online as well. Other than that, you can write to us through the website, and we'll be happy to respond to you and see how we can help you in testing your samples. We'll also guide you in case you have to come all the way to us if you're placed somewhere else in this country. There are other facilities which can help you out as well, but it'll be a pleasure to help in any way possible as far as the institute is concerned.
So the next question is from Ambarish Raj Deka. He's asking, is there any possible solution to the environmental problem to replace traditional plastics with bioplastics reinforced with nanoparticles?
Yes, so let's not put nanoparticles in everything, but I'm saying that people are already working again for bioplastics. People are using different kind of material since several years, including cornstarch, which are biodegradable plastics to a large extent. And I think that work is going on in a big way. And perhaps you'll also in CSIR, and perhaps you'll see success as we move forward.
Thank you, sir. Can we ask some more questions? Actually, sir, there are a lot of questions. So
up to you. I can't say
okay.
no. Being the host, I can't say no.
Lisa, that's the one consolidated. Okay. One final question.
Okay, sir. So one final question. Okay, sir. So the last question is from Madhupriya Selvaraj. How the cancer cell is formed in the human body? Which food mainly causes the cancer? Can we modify the DNA to remove the cancer cells completely, or by using xenotoxicity to remove cancer cells?
Wow, that's the entire cancer biology into one question. So let me just say from the other side, the easiest thing that I can perhaps take on, because the other things will take another lecture to explain how the cancer cell is formed. I mean, there are mechanisms in place where the DNA gets damaged and you know the programming of the cell goes bad and it starts replicating. That is one, or starts differentiating without stopping. So that is one. But on the other side, I think in terms of food, people have shown that not only this polycyclic aromatic hydrocarbons, if they're there in food, at some point in time when you over-grill things, especially non-vegetarian food, there are certain carcinogens that go into your food. Thirdly, I think what people say is that when you talk of water, you should be careful. There are certain pesticides that are there. Smoking, of course, tobacco and smoking is another thing which is always highlighted on the internet or on your social media, on television, that smoking kills because of cancer, and so does tobacco, chewing tobacco. So I think as long as we have good quality, enough of antioxidants, I think we are good to go. Vegetarian diet, I'm not for one saying that don't eat non-vegetarian food, but at the same time, we should also have large amounts of vegetables so that it controls the oxidative mechanisms that are taking place, and these antioxidants help you in a big way. So I think that is where I'll stop. And but I think it's a good question, that the fact that she has got these thought processes is good. But perhaps on another day we can discuss at length on cancer biology.
Sir, our students have prepared a slide based on your talk, so they just want to share it now. So you can have a look, sir.
Sure. Well, that's wonderful. They redefined CSIR.
All right.
Great. Fantastic. So this is something that I've been enjoying. These students are very creative, very innovative. That by the end of the lecture, they already have the crux of the lecture. Yeah. Fantastic.
They are next generation, much smarter than our generation.
Yeah, for sure, for sure. They think very quickly and they act even more quicker. That's quick. That's really good. Thank you very much. Whoever has done it, my compliments to you.
Yeah. Our student Lisa and Monty and Rajiv, they
Great
have
job.
done it.
And Lisa.
So thank you very much, sir. Now I request Miss Monty Gogoi to offer vote of thanks.
Thank you so much, sir, for your very informative, innovative talk. We got a lot of appreciation comments for your talk, sir, and we would like to hear more from you, sir. I would also thank our Sastry sir, who gave us the opportunity to prepare the slide for you and also to be a part of this program. Thank you so much, sir.
Thank you, Monty. Thank you. All right, Shastri, thank you so much. And I see you tomorrow then.
And we thank all the participants also for joining us for this talk. And some of the participants are joining through YouTube as well as Facebook Live. So with this, we end our this Eminent Scientist lecture. Thank you. Thanks a lot. Jai Hind.
Thank you. Jai Hind. Bye-bye.
Sir, Sastry sir, would you like to say something?
No, I am very much happy and delighted to have this lecture. And whatever has to be said has been said. The redefinition that Professor Alok Dhawan always tried to give it to CSIR has been very clearly portrayed. And let us work towards that. Let's make ideas are important and execution is more important. And we will resolve with much more dedication that we will work towards the glory of our country in this COVID times. Thank you very much. Thank you, Dr. Alok Dhawan, for exciting lecture.
Thank you.